Okay, so yeah, I was going to. I had the title I had, which was about TLR9 and BTK, but I think I've actually sort of changed the title slightly. It's a bit wordy, for which I apologise. But I'm going to talk about the mechanistic basis for disseminated aspergillus in patients receiving the tyrosine kinase inhibitor ibrutinib. I think the haematologists in the room probably know what I'm talking about, but effectively this talks to this area where there's a lot of patients who've been receiving ibrutinib for CLL primarily, and some of these patients seem to be getting disseminated, including CNS aspergillus, and it's a very interesting area that happens to serendipitously coincide with some work we've been doing. So it's a rambling story. I'll try to move through quickly, but there's a fair amount of data. So I'm going to talk about immunity to aspergillus in organ transplant, then discovery of this new TLR9 BTK calcineurin MFAT pathway we discovered in macrophages, how that interacts with this ibrutinib story, and then the effects of ibrutinib on human macrophages. So we're interested in aspergillus in the lab. We're in particular interested in what happens in the lung. This is aspergillus within a macrophage, and you can see here that this is what you don't want to happen. The aspergillus is germinating. That macrophage dies, another macrophage comes in and tries to control it. But ultimately, these macrophages can't, on their own, control the infection. This doesn't always happen, and we're interested in the mechanisms that lead to either control or progression of infection in macrophages, because it's the first event, or one of the first events in the lung. I initially got involved in the immune system, having previously been much more interested in the organism when I was training, and we showed that organ transplant patients don't make a very good TNF-alpha response when they have invasive fungal infection compared to healthy controls. And from that, I got funding to look into this in more detail. We're particularly interested in the role of steroids and calcineurin inhibitors, which are the main organ transplant immunosuppressive drugs on pulmonary aspergillosis. And we showed that combinations of steroids and calcineurin inhibitors like FK506 synergistically increase mortality in animal models. We showed that the calcineurin inhibitors, as well as the steroids, were having an effect. Calcineurin inhibitors such as FK506 impaired fungal clearance, and there was increased weight loss in a calcineurin inhibitor-treated animal, although these mice didn't succumb to infection. In parallel, we showed that within the airway, the calcineurin inhibitors seem to block all of the main cytokine and chemokine responses to fungal challenge intranasally, and that this was associated with a very impaired neutrophil recruitment early on in infections. The calcineurin inhibitors blunt your neutrophil response, neutrophil recruitment in the lung for some reason. We replicated this in a zebrafish model. I don't know if you can see that so easily, but you see a zebrafish with some fungi within the brain. And those animals, when treated with FK506, had increased mortality. And we were also able to show in whole zebrafish larvae that there was effectively very poor neutrophil recruitment to areas of infection, resulting in progressive hyphal germination. So based on those studies, we then wanted to understand how the calcineurin pathway is being activated in the innate immune system and what the mechanistic basis was for that. We initially had a look at which cell types were taking up the fungus, and using image stream, we were able to show that most of the fungus earlier on, when we had this clear phenotype in the airway, was landing up in the macrophage, at least by bronchoalveolar lavage. And we went on to show that if you silence calcineurin in these macrophages, you completely lost this TNF-alpha response, and that this was basically reproducible either with FK506 or gene silencing, showing that it's a sort of on-target effect for calcineurin inhibition. We also showed that aspergillus phagocytosis leads to activation of this calcineurin MFAT pathway, which the calcineurin inhibitors target. So you get blockade of the translocation of this MFAT transcription factor into the nucleus, and this can be blocked with FK506, the calcineurin inhibitor. So we concluded that tacrolimus FK506 inhibits this aspergillus-dependent activation of the calcineurin MFAT pathway in macrophages, leading to impaired TNF macrophage responses and subsequent defective neutrophil recruitment. So we wanted to understand how the calcineurin MFAT pathway is activated in macrophages, because usually it's activated in T cells and B cells. That's where this pathway is well recognized in the adaptive immune system, and it's less well recognized as a key player in the innate immune system. And we were aware that there was a very wide range of toll-like receptors and C-type lectins, as well as other damage and scavenging receptors, which are involved in sensing fungi. And so we needed to make some decisions about how to screen for activating receptors for the calcineurin MFAT pathway. We initially took the approach of looking at dectin-1, which is a sort of primary candida C-type lectin receptor and phagocytic receptor and signaling receptor. And we saw that, that this was important for both signaling and so was the MID88 pathway, which is a downstream adapter in that pathway. However, when we looked at NFAT translocation rather than the overall TNF response, it seemed that whatever pathway was being activated by Tectin-1 and MID88 seemed to be independent in Aspergillus of the Dectin-1 MID88 pathway. Whereas for zymosan, which is basically Candida or yeast, that pathway is important. So the zymosan activating calcineurin MFAT pathway is Dectin-1 and MID88 dependent, but for Aspergillus it seems to be a different pathway. We then went on to look at spleen tyrosine kinase, which is a tyrosine kinase which is downstream of about 95% of the C-type lectins. So we took that out either by inhibition or siRNA, and again we found that this didn't seem to be important for activation of calcineurin MFAT. So again, this pathway seemed to be both phagocytosis dependent and independent, of C-type lectin signaling. So from there, we went on to look at this relationship with phagocytosis that we'd seen on the confocal microscopy. And when we blocked phagocytosis, we got very clear loss of the calcineurin MFAT activation pathway. And there, interestingly, seems to be increased NF-kappa B translocation, suggesting that that does primarily come from the cell surface, whereas the MFAT pathway is phagosomal. And at this stage, we were thinking about intracellular signaling pathways that could activate the calcineurin MFAT pathway. And there have been some data to suggest that TLR9 was recruited to Aspergillus phagosomes, as you can see here, and that Bruton's tyrosine kinase is also recruited to these Candida phagosomes. So we took the approach of looking at these two molecules specifically, and we're able to show using knockout or inhibition that TLR9 was important for the NFAT pathway. And in addition, we were able to show that BTK inhibition or siRNA did ablate this calcineurin pathway response to Aspergillus phagocytosis. Based on that, we were able to construct this model whereby phagocytosis of Aspergillus leads to activation of this calcineurin MFAT pathway in macrophages, which is dependent on both TLR9 and BTK, although we're not really sure of the interrelationship there, and that this is basically blocked by the calcineurin inhibitors. And interestingly, at this stage, we were wondering about BTK inhibitors, because when we've been looking for drugs to use for our assays, we'd noticed that some of them were in clinical development for hematological malignancies. But at that stage, we didn't take it further. We were doing some studies to replicate the calcineurin pathway story in human macrophages to show that this is conserved in humans. And during that time, we were doing some microscopy, and we noticed this curious lateral transfer phenomenon where you see that this Aspergillus is transferred from this macrophage to the other one. So you can see that down here on this group of stills. So this one very clearly moves across to this one, and this macrophage actually stops moving, which we thought was a clue that it could be dying during this process. Actually, it does. Yeah, so have a look at that one. That one's clearly in that macrophage there, and then it pings across to that one. Lateral transfer. So because the donor macrophage always seemed to stop moving after that occurred, we added in different compounds to work out if there was a cell death process occurring. And using propylium iodide, which marks cellular necrosis by DNA release, we were able to show that the donor macrophage always goes red after transfer, which is the propylium iodide binding to DNA being released during the necrotic event. So this is very clearly a necrotic event that seems to be associated with improved control of fungal germination in the recipient macrophage. It's almost like a Aspergillus transplant between macrophages. And we were also able to show that this was calcineurin dependent. So it's about 50% reduction of this effect when you added in calcineurin inhibitors or performed siRNA. So based on that, we coined a term for this form of cell death because it hadn't been described before, known as metaphoresis, where basically if you have phagocytosis and then successful germination, there's strong activation of the calcineurin pathway, which is, through events I haven't got time to talk about, involved in this transfer during cell death, which is all interrelated to the calcineurin pathway and phagosomal sensing. Following on from that, we started getting involved with the whole ibrutinib story because there are increasing reports of patients developing invasive fungal infection with ibrutinib. And ibrutinib basically completely blocks, it's an irreversible inhibitor of Bruton's tyrosine kinase, which is this molecule that's important for this intracellular signaling pathway. There was a paper that came out last year that showed that around 39% of patients in a high sort of dexamethasone ibrutinib treatment regimen for CNS lymphoma went on to develop aspergillosis. And very interestingly, a lot of this aspergillosis was in the CNS, and I'll come back to why that's interesting in a bit. So this is basically the target protein. So there are 5 domains for BTK, and ibrutinib irreversibly binds at cis481. A lot of our assays look at secondary autophosphorylation this way, and there's a whole range of different interacting proteins and kinases, etc., that are known. And then there's a load of them, including the TLR receptors, including TLR9, for which the interacting domain is not known. And this, you can't see very well, but this is basically the paralogous pathway in B cells, where co-activation of the B cell receptor and TLRs leads to activation of BTK down to calcineurin MFAT. So there's a nice sort of parallel comparability between what's going on in B cells and what's going on in macrophages for this pathway. So we looked in human macrophages. We showed that the phosphorylation of BTK occurs in response to aspergillus and is blockable completely with ibrutinib, and that BTK phosphorylation is also required for NFAT and NF-kappa B translocation, as shown with this study here. In addition, using blockade of with either ibrutinib or TLR9 with ODN-2088, we can also turn off the NFAT signaling pathway, suggesting both of those molecules are important. And in addition, we basically followed this up with siRNA to show that this was an on-target effect, because ibrutinib has got activity against a range of other TEC kinases and also SERT kinases. So it's known to have, it's basically quite a dirty BTK inhibitor. Interestingly, we were also able to show that using phagocytosis blockade with cytokeratin D, that the BTK pathway appears to also be intracellular, so a phagosomally dependent pathway. And in addition, we showed that TNF-alpha production was also reduced, which is what you predict from blockade of both the NFAT and the NF-kappa B signaling pathways. This is, I don't know if you can see this that easily, but this is just some confocal microscopy. Here you can really see very nice co-localization of BTK with a fungal conidium that's germinating within a macrophage by confocal microscopy. There's this interesting issue that there's a load of patients, XLA patients, who've got mutations in BTK who do get progressive bronchiectasis, and they have a small but definite risk for fungal disease as well, but it's nothing like as severe as in patients receiving ibrutinib with steroids who've got pre-existing hematological malignancies. So we started looking at TNF-alpha responses in some of these patients. We've only done 2 thus far. The nice thing is that they've all got their genomes done, so we know where their mutations are. And thus far, interestingly, those 2 patients don't seem to have a defect in TNF, which is probably for very complicated molecular reasons, which I haven't even begun to investigate. But we're essentially going to try, potentially with Chris Eads, who I think is in the room, to go through a whole load of patients and look at whether or not mutations anywhere in this gene lead to a defect in the aspergillus signaling pathway in macrophages, to try and work out if that might give us an idea of where TLR9 is basically ligating into this pathway. So there's some interesting work to be done there. I just want to finish up with this slide because, or actually there's another one after this, because essentially what we found was that if you put ibrutinib or TLR9 blockade into a cell death assay for macrophages infected with aspergillus, you can effectively massively reduce the cell death response. And this got me thinking, because when we were developing these assays to look at this macrophage release of aspergillus, we were very mindful of a phenomenon known as vomocytosis, which occurs for Cryptococcus. And the interesting thing about vomocytosis is that what happens is that Cryptococcus is taken up by the macrophage, and then it's able to exit the macrophage by expulsion. But that expulsion event is not associated with death of the macrophage. And it's been hypothesized that Cryptococcus can be taken up by macrophages that then migrate to the CNS where the expulsion event occurs. And that's the mechanism by which you get CNS infection with Cryptococcus. So what we're now wondering is whether or not ibrutinib-dependent rescue of macrophage suicide responses drives the Trojan horse effect, which is what this has been termed, because there's a huge preponderance for these patients on ibrutinib to get CNS infection. It's way higher, and also disseminated infection. I think Samir had one in a joint with no white cells and things like that. Lots of strange things going on. So this is essentially what we're looking at the moment, but I haven't got good enough data yet to sort of show any of it in this setting. So just concluding this piece, rapid expansion of inhibitors targeting B cells and T cell signaling in transplant cancer and autoimmunity. And I mean, the hematologist can tell you about this, but it's actually, there's just a bewildering array of molecules now coming out, all targeting these pathways, which are supposedly only T cell or B cell specific, but actually, as to my crude understanding of evolutionary immunology, that whole adaptive immune response all basically evolved from the innate immune response. And I think there's a game that's being played that perhaps, you know, when these drugs are brought to the FDA and their mechanism of action is discussed, that potentially people are saying, oh, well, they're all specific to these cells, but actually they've got lots of sort of quote-unquote off-target effects on innate immunity. There is going to be a high rate of off-target effects on innate immunity, and lots of these patients getting lots of infections, probably all of which can't be explained just by B cell or T cell effects. There's this paralogous TLR9 BTK calcium MFAT pathway in the innate system, which seems to be important for aspergillus, and obviously the ibrutinib takes out the BTK component. Calcium inhibitors like tacrolimus, cyclosporin take out calcineurin. And there's this emerging story of BTK cell death and the CNS infection, which, you know, we've got a few sort of theories about, which I think are going to be really interesting to investigate. Just want to acknowledge Suzanne Herb, Sanan Shah, Amelia Berkeson, who did most of this work, Delia Warris, David Lowe, Serge Mostovi, who supported this, and the Imperial Fungal Group. Thank you very much.